Institute webinar series. This week we have Stuart Roy speaking. Stuart is an Associate Professor based in the Plant Genomics Group in the School of Agricultural Food and Wine at the University of Adelaide. He is currently the Director for the ARC Industrial Transformation Research Hub for wheat in a hot and dry climate. His interests are improving the abiotic stress tolerance of cereal crops, and particularly salinity tolerance, and improving cereal yield. Today he'll be speaking about high value genes for enhancing wheat yield. Just a reminder that to ask questions, the questions will be asked at the end of the session, and please type them into the Q&A box at the bottom of your screen, and I'll hand over to Stu now. Thanks, Stu.
Great, thanks Rebecca. Thanks for the invite to present. Rebecca, can I just confirm that everyone can see my screen?
Yes, I can see your screen, thank you.
Bro, great. Okay, so this is kind of an update on a presentation I gave at this time last year, which is around an international wheat yield partnership project that we've had for 3 years. In fact, this project I'm describing, the work has effectively come to an end. We're still ticking things over, but we do also have a new project that started at the beginning of this year, and I just want to take you through some of the results from the first project, and also where we're going with this in the future. So we're looking very much at wheat. In this, it's obviously an essential crop. It's the most widely grown crop globally, and sort of makes up for about 21% of the food calories and 20% of the daily protein in people's diet. The project with this wheat's focusing on enhancing yield under optimal growth conditions. And that's due to trying to make sure that the future demands, that we're in a good place for having enough wheat for making the future demands from the world's population. And this is just taken straight off the IWYP website. It's based on a sort of a graph that Peter Langridge did a number of sort of years ago. And it's on the prediction that if you've got a world population of a world population of 9 billion by the year 250, you're going to have to improve wheat yield production by 60%. And the current wheat trend is a sort of year as 1 to 1.5% increase per annum. But if you're wanting to make this 60% increase and sort of up this trajectory here, then you're going to have effectively a stepfold increase in yield, 2.5 to 3% per annum. There's concern that with diseases, with droughts, with climate change, etc., that yield could actually drop off. And so the IWYP program was designed from the Wheat Initiative, it spun out from the Wheat Initiative, and that was looking at, can you investigate ways of enhancing wheat yield by 50% in the next 20 sort of years or so, so a 2.5% increase in yield. The program's designed to bring together researchers from around the world with different funders all contributing to a project with multiple sort of collaborators on that. So researchers from Australia, the UK, Canada, America, Mexico, etc., and bring them together so that they can have a large project focusing on yield and yield enhancement. The program itself tends to have a range of different strategies and the types of projects that they fund. The early projects were very much based on, let's fund 10 to 12 high-risk projects in the hope that 1 or 2 of them will actually deliver the step-fold change, or that you might have several projects that can, with their inputs, join together to form a larger project that that does the step change itself. They're based around sort of utilising new technologies and building up collaborations. And a key part of the IWYP programme is actually in the delivery of germplasm or tools that can be used in breeding programmes. IWYP itself has what they call the IWYP Hub, which is a group of people that are based at CIMMYT in Mexico that evaluate the material from the IWYP programs and then look at transferring the material that's working out well actually into CIMMYT's breeding program, so it can then be distributed around the world. We've had a number of IWYP projects actually from the University of Adelaide. So I've led one. I'm now on another one. Matt Gilliam and his lab has been involved with an IWYP project led from ANU, and Scott Bowden's got what they call an aligned project, where the funding doesn't necessarily come from IWYP, but it's aligned to the IWYP goals and can make use of some of the facilities. So the project I'm going to mainly spend my time focusing on is this one here called IWYP39, and this project spun out from research that was done at the ACPFG and then sort of continued at the University of Adelaide and the University of Melbourne, and came from the focus in the ACPFG around looking at ways to enhance the tolerance of wheat to different abiotic stresses, or to look at increasing nutrient gain in the grain. Now, the IWYP program itself is not focusing on stresses, it's focusing on yield under optimal conditions. But what came out from the programs at the ACPFG is that some of the candidate genes we were working with and not only improved the tolerance of plants to stresses, but also had an unforeseen benefit on the growth of plants under optimal conditions. And so the IWYP39 project was looking at some of these candidates, which could actually be used to improve growth under sort of more optimal conditions. There's 3 genes focused in this study. The first of these was the vacuolar proton pumping pyraphosphatase gene, AVP1 from Arabidopsis, which, when you look at the literature and from research we did here in Adelaide, is reported to convey tolerance to multiple abiotic stresses. And we focused on salinity. This is some of Rhiannon Schilling's work from her PhD of transgenic barley plants growing in a saline field with the ability to actually yield compared to non-transgenic barley, which basically died. But the vacuolar proton pyrophosphatases have been implicated in salinity, they've been implicated in drought, in Arabidopsis, in nutrient deficiencies, transgenic rice having better growth under low P. And this initially was put down to the protein itself being on the tonoplast, pumping protons into the vacuole using pyrophosphate as an energy source. And then those conditions set up in the vacuole enabled either the compartmentation of nutrients or toxins into the vacuole, helping the plant sort of grow. More recent evidence, and where we started looking at this with the IWYP project, was there was also evidence starting to appear which suggested there was a role for this in sugar transport, and you might actually have better acclimation and distribution of carbon in these lines. The second gene came from Alex Johnson's work, which initially started at the ACPFG, and then he took with him to the University of Melbourne as part of his Harvest Plus program. And this is the cochianamine synthase 2, NAS2, which is an enzyme that helps in the formation of a metal chelator, the cochianamine. And Alex's work was very much focused on looking at enhancing iron and zinc in the grain of rice, and then more recently in the grain of wheat by overexpressing this this gene. And he found in his transgenic rice and also in his wheat as well that they were starting to get seeing sort of in green up here around the outside of it, more iron accumulating in the polished grain of rice and of wheat, and 4 to 5-fold increase in the amount of iron and zinc that you got in the grain. And so Alex has been pursuing this from a sort of more nutrition and a human health aspect, looking to try and get biofortified crops. So that's our cochianamine synthase. And the third gene came from Sigrid Hoya's work from when she was at Erie. And this was focusing on a gene involved in phosphorus starvation. Sigrid and colleagues mapped a QTL for growth under low phosphate conditions, and identified a gene, PISTL1, which is largely absent in irrigated rice, but found in rain-fed and drought-tolerant rice. And PISTL1, either in isogenic lines or in transgenic lines, they've got here, allowed the rice plants to grow under low phosphate conditions, giving them sort of enhanced sort of root growth and also biomass under those conditions as well. Why we were interested in these in the IWYP project was we started to actually get some anecdotal evidence or evidence in the literature that was always kind of skimmed over, that the transgenic plants actually perform better under optimal conditions as well. In terms of the vacuolar proton pyrophosphatase, work in our lab showed that in addition to sort of enhancing tolerance to multiple abiotic stresses, we're also seeing in the barley plants that Rhiannon was generating enhanced biomass and enhanced seedling vigor under optimal conditions. And when you went through the literature and read a bit deeper into the work done in rice and Arabidopsis, this phenotype was always there. It was just never emphasized in those papers. So the gene was enhancing growth under non-stress conditions. Worked with Alex in his field work when he was looking at his GM material in the field. He was also seeing evidence for increase in tiller number and sort of number of grain per head on his transgenic plants, along with his iron phenotype, but it was never properly characterized. And also worked from Sigrid when she was transferring the PISTL gene into wheat. In addition to seeing enhanced growth under low P conditions, she was also seeing evidence for improved biomass, potentially early vigor, but again, not characterized properly. And so where the IWYP project was starting was, let's get a better handle on this. Can individually these genes be used to enhance the growth and yield of wheat under non-stress conditions? But what happens when you start to combine these genes together, and you've got these proteins that do different things in the plant? You've got proteins involved in enhanced nutrient uptake, enhanced biomass of the plant, movement of sugars from source to sink tissues, etc. Could you come up with what was effectively a super wheat? What combinations of the genes work really well? Or if you did stick the genes together, did you just end up destroying the plant and the plant wouldn't function? So the goal of the IWYP project was to use GM plants as a proof of concept to see, can we use these three genes either individually or together to enhance the yield of wheat? And can we start to pull apart what are the mechanisms behind that? Now, given that there's no GM wheat released anywhere in the world and unlikely to be for the foreseeable future, when you knew what combinations of these genes work well, could you then find the wheat versions of these genes? Could you then look at was there any allelic variation in these genes? And could you potentially select select alleles of these genes that have got high expression, combine them into adapted germplasm and get a phenotype similar to approaching to phenotypes you might see in transgenic plants? And that would be passed on to the IWYP hub at CIMMYT. Or could we potentially investigate what's going on in terms of sort of genome editing? Could we use something like site-directed nucleogenesis 1, which the OGTR has deemed as being an approach that would not be regulated as GM? Could we use that approach to manipulate promoters of the wheat equivalents of these genes, knock out negative repressors and get enhanced expression of the genes, and then a phenotype similar to the transgenic plants? So that was the goals for this project. It involves a large number of people, as you sort of see at the end, but we had collaborators at the University of Melbourne, obviously Sir Alex Johnson, Sigrid Hoya when she went to Rothamsted Research, Julia Bailey-Series at UC Riverside, Roberto Gaxiola in Arizona State University, and then also Ravi Valura at who was our link to CIMMYT to get material out into CIMMYT. So in terms of assessing the GM plants and the proof of concept, and can these genes be used to enhance the growth of the plant, we evaluated this material in sort of the greenhouse and growth conditions in numerous labs around the the world, and also in the field as well, predominantly in Adelaide, but also some trials took place at CIMMYT in their GM facilities. So first step was to establish our 1, 2, and 3 gene combinations of our transgenic wheat. We did this by already having the 1 gene combinations, and to establish the 2, 3 gene combinations, we either did this by crossing the material together or by a direct transformation of wheat with the 2 and 3 gene combinations. The greenhouse evaluation took place in Adelaide, Arizona, and at UC Riverside, and the field evaluation predominantly in Adelaide, but also at CIMMYT's field trials in Mexico. And as part of this project, we generated over 137 different lines, different combinations of this material for evaluating either by the direct crossing or by transformation. And all stages, the plants we were comparing against were null segregant GM plants that had gone through the exact same history, the exact same processes as the transgenic lines. They'd just lost the tDNA insert that contained the gene of interest, and so effectively had reverted to wild type plants, but had the same environmental and epigenetic history as the transgenic lines. When I do say field trials, just so that we're sort of on the page, sort of really just talking about single plot trials. The GM trials were not large plots. We're working towards that now, but they were single plots or single row plots at our GM field site in South Australia. So our GM trials were done at Glenthorne Farm, which is just off Majors Road and Main South Road. And in terms of scales, you can get an idea of it. That's the area where we're allowed to do the GM trials. That's 1/4 of a hectare, and there's where the IWYP trials were done in there. So when I'm saying field trials, what I'm really meaning is something that was about 4 meters wide, 20 meters long, and single row plots. So just bear that in mind, that the field results are that, and they're not large plots. That's what we're working towards. So here's what our plots kind of look like in the field to give you an idea of the scale. So much smaller than what you would normally use, but they did actually allow us into the field, into the ground, and growing material in field-like conditions. They were always surrounded by buffer zones of wheat and of barley, and to avoid edge effects, and also as part of our regulation to control spread of pollen, etc., to make sure nothing escaped from the trials. In terms of yield and increases in biomass, we did identify some good genes and some good candidates, enhanced biomass and enhanced yield, particularly lines that involved either the ADP gene or the pistil gene. Just giving you an example of some of the results that we've got from this here, and if you want to follow up more, particularly about the AVP wheat, have a check out of the Rugami paper that came out earlier this year. The transgenics in the greenhouse always sort of put on much larger biomass than the null segregants, and had between sort of a 20% and a 65% increase in yield. And this was consistent in multiple trials in the greenhouse and multiple trials that took place in Adelaide, Arizona, and UC Riverside. In terms of translations to the field, we never saw the biomass phenotype in the field, possibly because this is more a water-limited environment. You're not wanting plants putting on such a large biomass, but consistently we were getting 20% yield improvements in these transgenic lines in those single row plots that we had out in the field. Similar story was seen with the pistil plants, an 18% increase in yield consistently in sort of the lines that were grown in the greenhouse, and between a sort of 9 and 21% improvement in yield when the plants were growing in the field. So there were definitely some good candidate lines that came out from that approach. When we're looking at combinations, the best combination of the genes that we put together were the triple combinations between the AVPs, the NASs, and the pistils. They performed really well and quite similar in the greenhouse conditions, less so in the field, a bit more variability in the field. The yield in those lines was 6% up to 30%, but again, promising material in there. And what we really want to do now is actually scale that up into larger breeders' plots and see what it looks like with less variability in there. Is this yield improvement seen at that larger scale? Some plants behaved well in the greenhouse and didn't behave well in the field. The AVP NAS and the AVP pistil lines had good yield and sort of biomass in the greenhouse, sort of seen here. But when you stuck them out into the field, they actually had lower yield in the field. And potentially, what we've done there is made plants have too much biomass that they struggle when there's water limiting, and that's something that we need to pull apart a little bit better. One of some of the material that we almost threw away, but was pulled up on by interactions with some of our breeders through the IWYP program, was the NAS and pistil material, which consistently had much lower biomass in the greenhouse, and also when we took them out into the field. But when they actually grew in the field, even despite having lower biomass, they performed fairly well with up to 19% improvement in yield in some of the lines in the field there. So they're actually producing more yield for less biomass, and for a rain-fed environment, that's something that we need to follow up on and explore that a bit more in detail. Unfortunately, we can't do GM field trials anymore at Glenthorne. It's been turned into a national park. You can actually visit the national park, and they've got wonderful plans for the national parks in terms of walking trails and things like that. You can currently get in and explore this area here. Our GM field trials are right across here, and we're currently in the process of decommissioning the field site there, so that it can be returned to a national park. But the silver lining from all of this was we got a new IWYP project and also interest from the university to build on their relationship with Sardi and actually start to develop a new facility for doing GM trials based at Sardi's Turretfield farm, and thanks to sort of the DVCR interested in putting up what's going to be 2 hectares of bird netting at Turretfield, which will allow us to actually scale up and do proper plots in these areas by people. Anyone that's got university project or external people can come in and use this facility for doing GM, but it's also going to be a facility that will allow material that's been produced by genome editing to go out there as well. One of the issues around genome editing you've got at the moment is that the OGTR has said site-directed nucleogenesis 1 will be considered non-GM, but the emphasis is on the researchers and the institutes to prove that it is just SDN1 and there's not any other little insert of tDNA that's been introduced accidentally into the material, because if there is, that's GM, and you can imagine trialing something that you thought was non-GM out on a GM field site, and a non-GM and a non-GM field site, and finding out it was GM under the definition by the OGTR. All sorts would hit the fan in that scenario, and so while you're actually proving that your material is SDN1 and is definitely definitely non-GM, you could actually be trialing that material out at the site at Turretfield to get some field trials and get some mileage before you can convert to non-GM trials. So that's on the go, hopefully built by the end of the year, and we'll start looking at using that facility in the near future. So we've got GM plants that are showing promising phenotypes in terms of yield in the field, and we then started to dissect what was going on with these trials, with these plants, and the mechanisms behind it. In terms of the AVP, we think this is very much a story around sort of improved source to sink translocation of sugars is one of the mechanisms behind this. These are called vacuolar pyrophosphatases, but it's becoming more and more evident that they have other roles in the plant and not just involved in sitting in the vacuole and pumping protons into the vacuole for setting up conditions for ion transport. Yes, they still do that, but there's other evidence appearing that they do other things in plants. One of them is that in tissue around the vascular tissue, they're not actually, well, they may also be on the plasma membrane, working backwards, actually synthesizing pyrophosphate, which can then be used as an energy source for sugar transporters to move sugar from source to sink tissues. And that's something that we looked at in non-transgenic wheat and in transgenic wheat in this project. So this is just having a look at an antibody that binds to vacuolar pyrophosphatases generally. So looking at binding to the wheat versions of the AVPs, so the TaVPs. And these are microscope sections from various cross sections of a plant. We've got leaf material here with concentrations of pyrophosphatase in the vascular tissue, again also in roots around sort of the phloem, etc. in the vascular tissue there. And so there is evidence that these pyrophosphatases can be concentrated around the vascular tissues. When you look at where these are in terms of the cell, and this is sort of amino gold labeling along here, the labeling is indicative of the native TaVPs in companion cells and sieve elements actually being on the phloem in there. And so that's again giving evidence to this theory that the vacuolar pyrophosphatases are actually helping with the sugar transport. We had a look at the movement of sugars from the shoots to the roots in the transgenic material. This is the wild type plants where you see the typical sort of movement of sugars from the source tissues to the roots, sort of peaking around about midnight and then dropping back down again. In the transgenic plants, this kept on going up. This is where we've effectively fed radioactive carbon dioxide to leaves and then tracking where that goes in the plants. It keeps on accumulating carbon dioxide overnight, and you end up resulting with transgenic plants with enhanced root biomass and also root length as well. We also looked at the transfer of carbon from flag leaf to the grain, and again, sort of the cross sections and staining for TaVPs found that you were having TaVPs around the vascular tissue in the flag leaf, and also in sort of around the epidermal storage cells and parenchyma cells and the peduncle in tissues there. And when you look at the transfer of radioactive carbon from flag leaf to the grain, the transgenics had more movement of the carbon from the leaf to the grain and to the florets as well. So the transgenic lines are moving more carbon from source to sink tissues. So we think that's one of the reasons why these AVP plants are able to produce more biomass and then also more yield as well. The pistil plants we've got evidence for sort of enhanced nutrient uptake in this. A lot of this work's been done by Julia Bailey-Seary's lab, by a master's student and a postdoc in her lab there. The pistil transgenic lines have enhanced sort of number of tillers on the plants when compared to the null segregant. You can see that they've got enhanced biomass, enhanced grain yield. They flower 5 days earlier than the non-GM plants, and RNA-seq experiments are showing that we've got a higher upregulation of genes involved in sort of nutrient uptake and movement of nutrients and ions through the plant. So we've got upregulation of genes sort of involved in sort of the mate pathway, metal transporters around sort of zinc and iron, nitrate transport, carbon transporters, etc. So we're seeing an upregulation of things that might enhance nutrient uptake and give us more biomass there. And Julia's lab's continuing to pursue these avenues of attack. Thanks to Jay at the PRC, we were able to have a look at what was going on in terms of the roots of the pistil plants, and the transgenic lines have, under low phosphate conditions, more of a rhizosphere acidification and more pumping of protons from the roots out into the rhizosphere. So here are the low P conditions. These are the null segregants, and then these are the transgenic lines, which have got a lower, or more acidic rhizosphere and more protons being pumped out from the root. This only happens at low P conditions. The transgenic lines, even though they're constitutively expressing pistil 1, there's obviously a regulatory component of the protein going on there, because at low sufficient P conditions, there isn't this difference between the transgenic lines and either rhizosphere acidification or sufficient P. So there's other processes going on here controlling the plant response there. So we're starting to get evidence for, we've got plants that can have higher biomass and higher yield. We're getting a better idea of what's going on in those plants. Can we make a non-GM version of this material so that we've got something that could be passed on to the IWYP hub, which could be then sort of included into their programs. So we had to first of all identify the TaVPs, the pistils, and the masses in wheat. Daniel Menadou, a PhD student in my lab, went and had a look through the new data coming out from the wheat genome, and identified 15 TaVP equivalents of AVP1. TaVP, so AVP1 is here. TaVP 1s and TaVP2s are the most similar to the AVPs, and there's 3 of these each. You then got TaVP3s and TaVP4s up here as well. So there are 15 of them around. And when you look at the expression, it's probably probably right to say that the TaVP1s and 2s are most similar to AVP1, which is expressed in vegetative tissue. TaVP3 is only expressed in the grain, but TaVP1 and 2s are expressed in the vegetative tissue. TaVP2D appears to be the most highly expressed TaVP in bread wheat, and it's expressed across a range of different cultivars. So this is just looking at some different accessions of bread wheat in roots, and different leaves of the plant, and sort of the sheath as well. So the TaVP2s constitutively in vegetative tissue, they tend to be more in shoot material rather than root material, whereas TaVP1s tend to be more root rather than shoot, but there's a bit of both. TaVP3s only expressed in the grain, and then TaVP4s are interesting in that they're either on or off in a cultivar. TaVP4 has sort of high expression in Scout and But Atlantico, but not in Mocho or Vigor 18, and so there's something sort of going on in there. The NASs with Alex Johnson and his postdoc Julian identified 21 NAS-like genes in the wheat genome, been able to get the chromosome position for 19 of these. 2 of them are suspected to be on chromosome 4 and sort of 3, and the closest 1 to NAS2 we think are those on chromosome 6A and 6D, and those are the ones we think are closest to the rice NASs that Alex has been working with. Pistil's been a bit of an interesting story, and we're still working with identifying the wheat equivalent of the rice pistil gene. Work with Uta Baumann and Yogi, identified 9 pistil-like orthologs in the new wheat genome that's been put out there. There was previously one pistil-like gene, you'll see why we call it pistil-like in a second, that was identified in wheat, which was this one up here on chromosome 5A. But when this was, when the new version of the wheat genome came out, this gene was found to be a low confidence gene. Work that we've done on it's found it's not actually expressed at all, and that Yogi Inuta pulled out 9 other pistil-like orthologs. The reason why we're calling them pistil-like orthologs are when you start to look at the actual protein itself, the rice pistil gene is effectively just a protein kinase, but all of the wheat versions of the genes we found, with the exception of the low confidence gene, have regions for transmembrane domains and wall-associated structures on their proteins, which is not seen in the rice pistil 1 gene. So whether these can function similar to the pistil 1 gene if they're actually on a membrane and in wheat, and the pistil ones floating around, and the rice pistil ones floating around in the cytoplasm, we don't know yet. And we're still trying to identify which one of these is the most closely related to the rice version. Obviously, the one that looks most like it is the low confidence gene that's not expressed, but that's not helpful. The pistil genes themselves, the rice pistil gene that Sigrid characterized is solely expressed in sort of the crown root. The wheat genes that we found so far are expressed all over the plant. We've got those that are expressed in the leaves, those are expressed in the root tip. The one that we think is the most likely candidate to the rice pistil gene that is not the low confidence gene in wheat is one on chromosome 6D. That's this guy here, which has got expression in the crown root and a high amount of expression in the root tip. This is this gene here, which has a transmembrane domain on it, but doesn't have any of the cell wall associated components. And so it's of the genes that are expressed, it's the one that's most closely related to the rice the rice pistil gene. So that's still work in progress. We did go out and have a look for allelic variation of all of the genes, and working with Delphine Fleury, and she had an Australian-India Strategic Research Fund. She screened 214 diverse wheat accessions, mainly bread wheats, but also durums and taushais and uratus in there. She had the 72 parents of the NAM population from sort of the wheat hub. And we screened for at the time, the known wheat versions of the AVP pistils and NAS genes. We sequenced the whole gene and also 2 kb of the promoter. We also got some data from Anthony Hall's group and their IWYP program, which is screening a high biomass wheat panel as well. So we've got some information about diversity there. We were able to find number of different haplotypes of these genes in the diverse panel. We initially split the genes up looking solely at their gene sequence and divided them up into groups based on haplotype groups based on that. But then recognizing that the transgenic plants have overexpression of the genes, so high expression of the genes, we started to actually form our haplotype clustering based on promoter analysis. So that we then start looking for genes with high expression in their promoters. And this is just a snapshot of some of the data that came from this in terms of the TaVP haplotypes. Sequence variation was 10 to 26% in the promoters, and we could divide them up into 4 to 14 haplotype groups per gene, depending on how the genes looked. This is an example of an output from the pistils. These typically could be split into 1 to 7 haplotype groups. We've got one of the examples of the 7 groups here. This is for the one that we think is most likely to be related to the rice pistil. These had a reasonable amount of sequence variation in there, and I've highlighted where some of the Australian cultivars sit in that haplotype cluster. Unfortunately, we couldn't do something similar with the NAS genes. The NAS genes themselves are so similar to each other, it's very, very hard to actually tell the NAS genes apart, let alone look for allelic variation within a NAS gene. And so we couldn't do this analysis on that at the moment. And Alex Johnson's now looking at genes that are further down the cocyanamine synthase pathway to see if there's more variation in those genes, and he's got evidence that there is for there. So we're having to take a new strategy for the NAS genes. When we've got the haplotypes, we're then able to go in and look at the different haplotype groups, split the plants up into different haplotype groups and look at the expression in these groups and start pulling out plants with differences in expression. This is looking at the AVP, or the TaVP2D expression profile. This is the gene that's the most highly expressed in bread wheat and vegetative tissue. And here are some Australian sort of cultivars here with reasonably high levels of expression, but we have accessions such as this Oz sort of line here in Bucket Atlantico with considerably higher levels of the TaVP expression. And what we're doing now is we're in the process of a back-crossing program of bringing in those alleles with high expression into Scepter. And we've been doing a number of sort of back-crossing bulking so far, so that we end up with Scepter with the allele for high expression of that, the TaVP or the pistil, and we'll be in a place soon to actually evaluate to see have we actually improved Scepter growth by incorporating these alleles in there. We're in the process at the moment of getting everything sorted out, so hopefully in the next month or so we'll be able to send to CIMMYT some of this material so they can start incorporating it into their programs at the Wheat Hub, so that they can evaluate that at CIMMYT and can also start to put anything that's useful that comes out from that into their breeding programs. So that's really what I want to sort of say about the first IWYP project. I just want to sort of make you aware of a second program that we've got going at the moment. Don't really have any results for it sort of yet to present, but just so you know what's going on. We've started at the beginning of the year a new IWYP collaboration, which has come about from an interaction between researchers Agriculture Agri-Foods Canada and at the University of Essex. And this is project designed sort of by them, was looking at stomatal signaling pathways and how you can manipulate either the stomatal behavior or patterning on in wheat, and see if you could either enhance yield or enhance water use efficiency, or could you kind of do both as that program. And so this is a project that's led by Nora Ford at Agriculture and Agri-Foods Canada. She's at Lethbridge, and Tracy Lawson at the University of Essex. And the Adelaide component was myself and sort of Penny Tricker, and we were brought into this project because of sort of the phenotyping skills and capabilities we'd have from the campus with like the accelerator, and also that we can do GM field trials and sort of experience in doing GM field trials, which are notorious for sort of setting up and running. So obviously looking at sort of stomates, because they're the main entrance of carbon dioxide for photosynthesis, and also involved in the release of water and oxygen from the leaves, and sort of maximizing sort of grain yield is going to be a balance between sort of maximizing the amount of photosynthesis you can have, so you can get biomass and get material up into the grain, but then also balancing water use efficiency. You don't want your stomates open all the time, otherwise you'll lose water, but then also maintaining optimal leaf temperature. You need to have your stomates open to let sort of heat out from the leaves. And this project here is looking at, can you manipulate stomatal conductance in wheat by subtle changes in either the anatomical features of the leaf in terms of density of stomata, or how they respond to environmental stimuli, and see, can you build sort of a wheat plant better in that way. And so the project from the Canadian and the British side is very much in understanding these processes and looking at how this can be manipulated, and understanding how stomates, etc., are formed. From our side of things, we're getting their mutant material and their transgenic material, and we'll be evaluating that in the field to see if any of those changes that they're doing actually affect yield of the plants. So there are a very large number of genes involved in controlling stomata development. A lot of the processes in stomata development are very well understood in dicots, less so in monocots. And we can't target all of the genes in these processes. So it'd been decided to focus on some very specific genes involved in either stomata development or in controlling stomata behavior. The genes focused mainly on the sort of development are the mute genes and the Yoda genes. Mute encodes for transcription factor, and that's sort of involved in the process of the development of sort of stomata, particularly subsidiary cell formation around the stomata. In monocots, you need these specialized subsidiary cells around the outside of stomata to actually sort of be part of that mechanism for opening and closing the stomates. And it's hypothesized if you knock out the mute, then you sort of knock out this whole pathway here. You don't get the differentiation of these cells into guard cells, and you don't get any stomata on these plants. The Yoda sort of protein is a protein kinases that's sort of involved in a number of different developmental pathways in that. And this has been sort of shown in sort of other species and things that if you've got mutants in the Yoda gene, what you end up with are clusters of sort of stomates around on the leaves. So this is why this has been included in the program. And for me, this has been a highlight because finally I get to actually start to work on a Star Wars gene. And so we're going to be working with what we effectively call in the lab baby Yoda plants. In terms of behavior, we're focusing on open stomate 1 and the SLAC1 genes. Open stomata 1, OST1, is a kinase which regulates ion channels such as SLAC1, and the ion channels are responsible for membrane depolarization, resulting in the activation of potassium transporters, and the stomates close. So this is a process involved in opening and closing stomates, and knockouts of OST1, they've got an open stomata phenotype, and likewise SLAC1 ion transporter, and again knockouts of these guys have open stomates. So the collaborators on the project, they're developing GM wheat with alterations in the expression of these genes, so overexpression or knockouts by genome editing. And we're also getting in EMS mutants for characterization in cadenza background, but also in Australian backgrounds, and looking to, while they're looking to sort of uncover more information about the involvement of the processes involved in stomata function and development in monocots and in wheat, we're going to be looking at are any of those modifications they've been doing actually resulting in enhanced yield and or water use efficiency. Pre-COVID, our role was very much in bringing in cadenza mutants from the sort of the UK, and we have brought in 44 lines currently going through quarantine, and we're genotyping and phenotyping those as we go along. We're already starting to see some nice mutants in those lines, and we would then send a postdoc to the University of Sydney to learn some of the approaches they use there for doing tilling of mutant populations. There was an agreement, I believe set up with Peter Langridge a number of years ago between the ACPFG and University of Sydney to produce some tilling mutants, and those will be the mutants that will be screened in this project. And the idea was to send a postdoc there to bring back some mutants, but also to bring back Sydney's tilling technique to the lab. And then we'd also bring in the transgenic and genome edited plants that were created in Canada and the UK, and then ultimately phenotype using facilities like the Plant Accelerator and the GM sort of field site to investigate whether or not we've been able to improve the yield of the plants in both optimal and drought conditions. Thanks to COVID, the project's taken a little bit of a hit, and we're having to frantically reschedule and rejiggle things, and it's been a bit interesting in trying to balance it all. We are still able to characterize and bring in the cadenza mutants. That's happened. It's just happened a bit delayed because the UK couldn't send the mutants across to us, but that's going at the moment. Our whole plan to send a postdoc to Sydney took a double hit because our poor postdoc is stuck in the Netherlands and can't get back into Australia. And the person across from the University of Sydney was effectively out of a contract at the end of August. And so we've had to subcontract the tilling work to be done at Sydney to keep their expertise there, to generate the lines and to send that material across to us. And the Canadians and the British have taken a hit in that they can't get in their labs to generate any of the GM plants. So they're now sending us their constructs that they've produced, and we'll generate the transgenic wheat plants here in the next couple of months using their constructs, and we'll do bialistic transformation on that. So sticking with the Star Wars theme, we had an option of either proceed with this project and do it, or to give up, and so we decided to go ahead and do it, to do or not do, and there is no try, so we're definitely going to do it. So that's where we're going with that the project at the moment. So summary, from the first project, we've generated transgenic lines that have in single row plots a 30% yield enhancement in the field. We're going to now evaluate that in larger plots. We do have enhanced phenotypes due to better sugar remobilization and sort of perhaps better access to water and to nutrients in the lines. We do have non-GM alternatives being developed using high expression alleles of the TaVPs and pistils. That still has to be checked and phenotyped, and we're going to be flicking that material across to CIMMYT so they can evaluate it there. And we've now got a new IWYP project looking to manipulate yield and water use efficiency underway. This has been a cast of thousands involved in this across a number of different institutes. Each institute has its own bit of funding to come into that. I've got to acknowledge the GRDC, who have funded both projects, and the Australian component for my side. Project 1 had funding from USDA, USAID, as well. And the new project we've got, GRDC, Agriculture Agri-Food Canada and the BBSRC, and it's all been brought together by the Wheat Yield Partnership. I'd like to particularly thank Yogi, Laura, Christine for a lot of the bulk of the grunt work of phenotyping, and Jan and Melissa for keeping us out of trouble in terms of GM, and the new group here, sort of Penny and Abdel on the new project. And at that, I'll thank everyone for their attention and happy to take questions.
Thank you very much, Stu. We've got a few questions. If you want to ask a question, please type it into the Q&A box at the bottom of your screen. So the first question from Eileen Scott is, are there any plans to examine the effects of root zone acidification in the pistil on mutant on rhizosphere microbiota?
So we haven't, not specifically for that, but we are having a look at the potential implications of the enhanced acidification in the plants. One of the issues that our collaborators had at UC Riverside was that they started to develop a very odd stunted phenotype, which when they showed us is a classic symptom of too much acidification around the roots, and the same kind of phenotype you see in wheat plants when they're grown in very acid soils. We were actually seeing in these transgenic plants, and effectively the transgenic plants had made the soil they were using, which was already slightly acid, too acidic, and they were struggling there. So we've certainly got to look into that, because that would be potentially an issue if because we've already got problems with acid soils here.
So Victor Sadras has asked, what is the genetic background where transgenics increase yield 30%?
Yep, so that background is predominantly fielder, which is a US cultivar that is very easy to transform by agrobacteria. That's why we've always compared to sort of the fielder nulls, because it's not, you know, in terms of, it grows all right in Australia, but it's not best. It's why when we're doing the non-GM stuff, we're wanting to actually bring things into things like Scepter, which is better adapted for the environment, so we can actually have a look at, has any of the results we've generated actually relevant to Australia?
Okay. Penny has asked, if lines have improved sugar remobilization, why don't they still have improved yield when water is limiting? Any ideas?
I'm not sure if Penny's allowed to ask any questions. It's something where we're, the biomass plants did put on a lot of biomass in the greenhouse, and it looked like they were trying to do the same in the field, but we had in the years that we're out in the field, we had very low rainfall, and none of the GM trials are irrigated. It's all naturally rain fed, and it looked like they were just struggling to bring up any water into the plants in those conditions. We did have a crack at trying a drought experiment in the accelerator with some of the material, and while we did actually see the plants behaving properly, well, not behaving properly, behaving consistently in that they were producing sort of more biomass and things, we couldn't get a drought effect happening in the accelerator, even though we did try droughting them. So that's still an open question that we need to investigate further.
Victor has another question in relation to the mechanism of growth enhancement. To what extent is leaf area and light interception, and to what extent photosynthesis per unit area?
Yep, so we haven't pulled that apart. There are other IWYP projects looking at that in terms of interception of light and photosynthesis. So Richard has got a project looking at light interception, and Barry Pogson, the project Matt's been involved in, has been looking at sort of more energy and photosynthesis around that area there. So we haven't looked at that specifically in our plants, and we were just trying to see if we could get effect first of all, and now we need to start pulling things like that apart to get more answers to that. So I don't have an answer to Victor. It's something we need to look at.
Okay, thank you, Stuart, and that's it for our questions today. So thank you very much for presenting. And I'd just like to remind everybody about the speaker that we have next week. Next Thursday at 11 o'clock is Dr. Stephen Lappage from the Fight Food Waste CRC, and he'll be talking about fighting food waste for a sustainable food future. How is Australia performing? So I hope to see you all attending next week. So thank you, everybody. Thanks, Stu.
No worries.